For decades, one of the biggest questions in all of science was, deceptively simple. What are we really made of? Not ourselves, not our tissues, but the actual instructions. What is the molecule that holds the blueprint for a flower or a mouse or you and me? So let's dive into the incredible scientific detective story that finally solved biology's greatest mystery. So the central question, the one that drove this entire search, was simple but so profound. What molecule carries the blueprint of life? I mean, scientists knew something was being passed from parent to offspring, carrying all the traits for everything from your eye color to your height. But what was this actual physical stuff? The hunt was on. By the early 20th century, the investigation had pretty much narrowed it down to two main suspects inside the cell's nucleus: proteins and this other thing, deoxyribonucleic acid, or DNA. And you might be surprised to hear there was a clear favorite. Everyone's money was on protein. It just made sense. Proteins are incredibly complex, built from 20 different amino acids. It's like a language with 20 letters. DNA, on the other hand, just seemed, well, kind of boring. It only had 4 repeating units. It seemed way too simple to hold the massive library of information needed for life. But, science is all about following the evidence, even when it leads you somewhere weird. And in 1928, a British physician named Frederick Griffith stumbled upon a clue that was about to change everything. It wasn't even what he was looking for, but it pointed the entire investigation in a radical, totally new direction. Okay, so check this out. Griffith was studying pneumonia bacteria. There was a deadly version, the S strain, and a totally harmless version, the R strain. Pretty straightforward, right? And just as you'd expect, if you killed the deadly S strain with heat, it couldn't hurt a mouse. But then, here's where it gets wild. He mixed the live harmless R strain with the dead S strain and injected that combo, and the mouse died. Something from the dead bacteria had literally turned the harmless bacteria into killers. Griffith called this mystery substance the transforming principle. He had proved that genetic information could somehow be transferred from dead cells to living ones, but he had no idea what molecule was doing it. So he'd found this crucial piece of evidence, but honestly, the mystery just got deeper. The chase was now on to figure out what on earth is this transforming principle. For almost 15 years, that question just hung in the air. Then, a team of researchers at the Rockefeller Institute, Oswald Avery, Colin MacLeod, and Macklin McCarty, came up with this brilliant experiment to methodically hunt down and finally identify Griffith's transforming principle. Their approach was just a masterpiece of methodical elimination. They purified the transforming stuff, and then they treated different samples of it with enzymes that were designed to destroy just one specific type of molecule. So, first up, they destroy the protein. Did transformation still happen? Yep. Okay, so it's not protein. Then they destroyed the RNA, and transformation still happened. But then when they finally used an enzyme that specifically chews up DNA, bam, the transformation stopped cold. The harmless bacteria stayed harmless. Their conclusion, which they published in 1944, was absolutely revolutionary. This was the first really powerful experimental evidence that DNA, not protein, was the carrier of our genes. But, old ideas die hard. A lot of the scientific community was still super skeptical, still convinced that this simple little DNA molecule just wasn't up to the job. The world needed one final, undeniable piece of proof. And that final proof? It arrived in 1952, with this elegant and now famous experiment by Alfred Hershey and Martha Chase. They used a type of virus that infects bacteria, called a bacteriophage, and get this, an everyday kitchen blender to settle the debate once and for all. Their genius was in how they made the invisible visible. See, these bacteriophages are basically just a protein coat surrounding a core of DNA. They work by injecting their genetic material into bacteria to make more copies of themselves. So Hershey and Chase cleverly made 2 batches. In one, they used radioactive sulfur to tag only the protein coats. In the other, they used radioactive phosphorus to tag only the DNA. It was like they put a little glowing tracker on each of the 2 suspects. The experiment itself was so beautifully simple. First, they let the radioactive phages infect the bacteria. Then, they put the whole mixture in that blender to literally knock the empty virus shells off the outside of the cells. Finally, they used a centrifuge to spin it all down, separating the heavier bacteria from the much lighter virus coats. And this all led to the million-dollar question: where did the radioactivity end up? Which one of those glowing tags would be found inside the bacteria, and which one would be left floating outside? The answer would reveal without a shadow of a doubt the true identity of the genetic material. And the result was definitive, just crystal clear. The radioactive phosphorus, the tag that was on the DNA, was found inside the bacteria. The radioactive sulfur from the protein coat, it was found outside. DNA was the molecule that actually entered the cell to give the instructions. It was undeniable. DNA was the genetic material. Case closed. So the mystery of what the gene was made of was solved. But like great science always does, this answer just led to an even bigger question. How did the seemingly simple molecule actually work? How could it possibly store so much information and get copied so perfectly? The answer, it turned out, was hidden in its structure. Now, scientists already knew the basic building block of DNA was a thing called a nucleotide, which has 3 parts: a sugar, a phosphate, and a nitrogen-containing base. And they knew there were 4 different bases: adenine, guanine, cytosine, and thymine, or, A, G, C, and T for short. But how all these little pieces actually fit together, that was a complete mystery. A huge clue came from a biochemist named Erwin Chargaff. He analyzed DNA from all sorts of different species and found this really bizarre but incredibly consistent pattern. In every single sample, no matter where it came from, the amount of adenine was always equal to the amount of thymine, and the amount of guanine always equaled cytosine. A equals T, G equals C. This became known as Chargaff's rule, and any correct model for DNA would have to explain it. The final pieces of the puzzle all came together in 1953. Using these critical X-ray images produced by Rosalind Franklin and Maurice Wilkins, 2 guys named James Watson and Francis Crick finally cracked the code. The structure was this elegant double helix, like a twisted ladder. The sugar and phosphate parts formed the outside rails, and the bases faced inward, forming the rungs. And this is where it all just clicked. A on one strand always paired up with T on the other, and G always paired with C. This perfect pairing didn't just explain Chargaff's rule, it revealed the beautiful secret to how the molecule could be copied so precisely. Unlocking the structure of DNA didn't just solve a puzzle. It gave us the ability to read the blueprint of life itself. And that double helix structure, it's not just elegant. It gives the molecule an almost unbelievable stability, allowing it to preserve information across thousands of years.
I mean, how stable are we talking? Well, get this. In 2010, scientists sequenced the entire genome of a man who lived 4,000 years ago in Greenland. They nicknamed him Inuk, and his DNA was pulled from tufts of hair that were found preserved in the permafrost. The information stored in that double helix was still perfectly readable after 4 millennia. It's just incredible.
And the level of detail they could pull out was just astounding. By reading his genetic blueprint, they knew he likely had dark skin, brown eyes, and blood type A positive. They could even see that he was genetically predisposed to baldness. This isn't just abstract data. It's a person's story, written in a molecule that survived for thousands of years. So we went from a simple question about what carries our traits all the way to the double helix, a molecule connecting us to our most ancient ancestors and giving us the tools to actually shape our future. The discovery didn't just close a case, it opened up a whole new world of possibility. And it leaves us with this one profound final question. Now that we can finally read the blueprint, how will we choose to write what comes next?